This is GATE 2022 Life Sciences Section P Chemistry. Question number 61. The bases present in DNA are A. Adenine, cytosine, guanine and thymine. B. Adenine, guanine, thymine and uracil. C. Adenine, cytosine, thymine and uracil. And D. Cytosine, guanine, thymine and uracil. So this is pretty basic and easy question for 1 mark. This is the NPTEL online course reference. So we all know the answer. The answer is A. In DNA we have thymine instead of uracil, and in RNA we have uracil as a base. Okay. We'll briefly go over the bases in DNA and RNA. Okay. Quickly. So in DNA, as we all know, we have adenine, guanine, cytosine and thymine. Okay. So bases are of two types, purines and pyrimidines. You need to remember this. So purine is a 9-membered ring. Okay, and this is how the numbering is. It starts from N1. Okay, 1, 2, 3, 4, 5, 6, and then N7, 8, 9. Okay, purines are the 9-membered ring. Okay, these are 9-membered ring. Whereas pyrimidine, as the name is longer, it has only 6-membered ring. Okay, you can remember like that. Purine has a shorter name, but it is 9-membered ring, whereas pyrimidine is a 6-membered ring. Okay, so there are purines are of two types, adenine and guanine. And in pyrimidine we have cytosine and thymine in DNA, and in RNA in place of thymine there is uracil. Okay, very basic. So in these bases we have a ribose sugar. We have a ribose sugar and we have a phosphate linkages. So ribose is attached to the N9 of purine. So here the ribose is attached, and at N1 position we have of pyrimidine we have ribose sugar attached. Okay, and at N3 here we have a phosphate. At N3 we have a phosphate, and here at N1 we have a phosphate. Okay, to form a nucleotide base. So there are apart from in DNA we have only 4 bases: adenine, guanine, cytosine and thymine. Whereas RNA we have different modifications of bases. So we have methylcytidine, N6 methyl adenosine. Okay, these are the different modification found on the RNA basis. Okay, N2 methyl guanosine. So approximately 5% of the 5% of the cytosine is modified to 5-methyl cytosine. Okay, and these modification essentially what they does, they protect us from various bacteria and viruses. Okay, so 5% of the cytosine of the human genome is methylated. We have 5-methyl cytidine, and this modification is mainly for the protection against various restriction endonucleases, various REs, which are released by different endonucleases which are released by viruses. So this help us, and bacteria. So this help us again protection to those restriction endonucleases. Okay. Apart from this we have ionosine, pseudouridine, 7-methyl guanosine and thiouridine. These bases are mainly present on tRNA. Okay, it is very important to remember that these 4 bases, ionosine, pseudouridine, 7-methyl guanosine and thiouridine, these bases are present on the tRNA. Okay, these are the modified RNA bases which are mainly found in the tRNA. Okay. apart from this, there are also questions on sugar puckering. The conformation, the sugar ribose and deoxyribose can have two conformation. One is C2 endo and other is C3 endo. Okay, so we have a ribose and deoxyribose. So the only difference is that at C2 position of ribose we have a hydroxyl group, whereas the C2 position of the in DNA it doesn't have any hydroxy. That is why it is deoxyribose, and ribose has a hydroxyl at C2 position. So generally C2 endo is found in. So there could be two conformations. So in this plane, in the plane of symmetry, this is the plane of symmetry. Okay, this is the plane of symmetry. The ribose could be either puckered inside, so we call it C2 endo. If it is puckered outside, if it is puckered inside, we call it C2 endo, and if it is puckered outside, we call it C2 exo. Similarly, the puckering is also observed in C3, right? So C3, if this is the plane of, this is the plane, right, this is the plane of the axis symmetry, and if it is inside the sugar, it is C3 endo. If it is outside, it is C3 exo. So in ribose has a C2OH, right? Ribose has a C2OH, and because of this C2OH is present in ribose, it will have a C3 endo sugar puckering. Okay, it will have a C3 endo sugar puckering. This C2 cannot move because of the OH present here. Okay, so this C2 OH will have a steric hindrance, and that's why we'll observe a C3 endo sugar puckering in ribose. Okay, whereas in deoxyribose doesn't have any C2OH. So the aDNA, so we have various forms of aDNA, bDNA and zDNA, right? So aDNA will have a C2 endo, right? aDNA will have a C2 endo conformation of sugar puckering, and bDNA will have a. So coming back to the question, the answer is adenine, guanine, cytosine and thymine. These are the bases found